Okay, we're going to start again. So we learned a little bit about gene organization and how genes are regulated. And now we're going to talk a little bit more about the kind of things that lead to evolution of bacteria, and that is mutation and genetic exchange. So this this leads to the evolution of virulence. You have mutations, which are changes in the DNA. These can be caused by mutagens, which are chemicals or physical agents that can damage the DNA. The cell is trying to repair the DNA, and it can repair the DNA very faithfully, so there's little chance of mutation, but there's also a system where it repairs the DNA and makes mistakes. And then we'll talk about the first mechanism of genetic exchange, which is called transformation. So mutation and recombination and selection, these are the things that are the driving forces for interaction with bacteria. So mutation is a change in the DNA sequence. A recombination, as we'll see, is a way of two pieces of DNA can exchange. And then selection is whatever we do in the environment that allows one organism to grow over another. Okay, so for example, if you add an antibiotic to a medium, that's a selection, right? Because any bacteria that isn't resistant is going to be killed if it's a bactericidal antibiotic, and one that's resistant can then proliferate. So, and then as we find out, these genetic exchange mechanisms, they allow transfer of genes between bacteria. And then genes that are favorable or confer an advantage to the bacteria are going to be selected for. And if it doesn't confer an advantage, then it's sort of immaterial. And by contrast to higher eukaryotic organisms, the nice thing about bacteria is the problem with bacteria is you're dealing with such large numbers of organisms, right? I mean, a generation time of 20 minutes, you start out with one cell of E. coli or salmonella or something. Within 10 hours, if there's enough media, you have 10 to the 9 cells. And as long as you keep providing media, the numbers will just keep increasing. So just think in 10 hours, you go from 1 to 10 to the 9. So bacteria can rapidly expand their numbers provided there's the nutrition. Okay, so selective pressure. So like I said, one kind of selective pressure would be addition of an antibiotic to a population. So that selective pressure is applied to total population, and then a mutant that's in the population that's resistant, in the absence of that antibiotic, wouldn't have any advantage, right? It would just be one of millions or billions. But as soon as you add that antibiotic, then you favor the growth of that resistant organism, and the other ones are prevented from growing. So that's going to lead to the emergence of a new population that is able to grow under that condition. So, but then the selective pressure could go away. You stop giving the antibiotic, and now the selective pressure is gone. The organism that's resistant doesn't have any advantage over the one that's not resistant, right? So it could disappear again. So here's an example. You have a bacteria, let's say, colored like this. It divides. Let's say a mutation has occurred, so this bacteria has one base different from this one somewhere in its DNA, and it divides, and all the progeny will be the same. All the progeny here would be the same unless another mutation occurs. And let's say this mutation confers resistance to a quinolone, one of the antibiotics that Dr. Reed talked about, and it's a bactericidal antibiotic. So this bacteria would be resistant to quinolone, the rest of them would be sensitive, so if you added the quinolone, the rest of them would all be killed, and you'd only select for this population here. Okay, so the addition of that antibiotic confers a selective advantage only to those bacteria that are resistant. The rest of them are sensitive, and so they're wiped out. So, okay, bacteria, are versatile. We've already talked about the regulation of gene expression, how they sense their environment, and then express genes or don't express genes that are going to be an advantage. But now we're going to talk about mutation and gene transfer. Okay, so If we look at mutations, what are mutations? I'm sure most of you are aware, are just changes in the DNA sequence, right, that are inherited from mother to progeny cells. And so what are the kind of things that are mutations? So there's base substitutions. Let's say you have an A and you substituted that with a C. That would be a substitution that would change the sequence, right? Instead of having an A there, you'd have a C there. Or there's mutations where you can add a nucleotide to the sequence or delete nucleotides. And in fact, you can have large deletions, although insertions are usually rather small, but except for transposable elements, which are like that vancomycin resistance is present on a transposable element. And a transposable element is a DNA element that can move from one location to another. So it could be on a plasmid, it can move to the chromosome, or it could be from the chromosome, it can move to the plasmid, and we'll talk a little bit more about that. So any mutation is a change in the genotype of the organism, right? So it has a different genotype, and usually some mutations are silent, but other ones alter the organism in some way by changing the expression of a gene or changing the function of that gene. So you could have a protein that is less active because you change some amino acid you change the sequence of the amino acids in that protein. It may be less active, or it may have a new function. For example, it could be resistant to quinolones. Or it may not work very well. If it doesn't work very well, the organism could die. So any mutation, and these kinds of mutations that change the expression or function of a gene is going to change the phenotype, right? So you could be quinolone sensitive, you could be quinolone resistant. Okay, so why are mutations occurring? And it allows the organism to change, respond to its environment, and have organisms that can survive under different environmental conditions. So organisms try to avoid mutations by and large, but mutations do occur. So in bacteria, there can be spontaneous mutations that occur just as a result of replication, or some chemical could be put in the environment, or some radiation, ultraviolet light, and these cause mutations. So those are induced mutations. And in bacteria, since they only have one copy of the gene, like we're diploid, so we have two copies. If one gene got damaged, you could still have the other one could be wild type. But in bacteria, they only have one copy. So if that copy is changed, it affects the organism. And so, as I said, then if that confers some selective advantage to the organism, that organism will survive at the expense of the other ones that are there. Okay, so what are the kinds of mutations? The spontaneous mutations, these occur naturally. It's the price you pay for having a molecule like DNA that has to get replicated, right? Bacteria have a chromosome that's 4 million base pairs long, like E. coli, and you replicate that, and basically it's pretty high fidelity process. In fact, physicists, before the discovery of DNA, could not figure out how you could have something that could be so faithfully replicated. But it turns out that DNA can be pretty faithfully replicated. And so that the probability that you get a mutation in a given gene is very low. Okay? And the low mutation rate is due to the fidelity of the DNA polymerase, and there's some repair mechanisms to ensure that the mutation rate is very low. Because most mutations are bad, and I guess if the organism didn't have this high fidelity of copying itself, the DNA of copying itself, you would have too many mutations. So base substitutions are the most common kind of mutation. And so, for example, if you have, up here in the DNA, if you substituted, you put a C across from an A, normally you would put a T across from an A, but if a mistake happens and you put a C across, then if that doesn't get repaired and it gets copied, then instead of having AT here, you'll have a GC in the other strand, or in the other copy. And so here, you have a triplet code, so here it's AAG that gives you lysine. Now, because you've put a G here, because when you've copied the C, you put a G there, now you have AGG and you put arginine there. So normally you have lysine at this position in the protein, now you put arginine there, that may have an effect, it may not have an effect on the protein. That kind of mutation is called a missense mutation, right? Because you've altered the sense. Instead of having lysine, you have arginine. There's also the possibility that you could make a mutation by base substitution that you would generate a stop codon. If you generated a stop codon, UAG or UAA, then the ribosome would come along, you hit that stop codon, you truncate the protein. That's probably going to be deleterious. So a nonsense codon, if you change a codon to a nonsense codon, you stop translation, and that's most likely not going to be very good for the protein because it would be truncated. Okay, so you can also have mutations where you add or remove nucleotides. This can also happen during replication, or it can also be due to some chemicals, as we'll see. So you can imagine this is even more deleterious because it would shift the translation reading frame. So you read the messenger RNA in a triplet, right? So here's an example. So here's the DNA strand, here's the RNA strand. So you would read this, you would have CGG would be arginine, UAC tyrosine. If you stuck an extra base in there, then now you'd read arginine, but now you'd change the reading frame. So instead of having UAC, the extra base would lead to AUA. You'd have isoleucine, and then arginine. Now here you hit a stop codon because you've shifted. Now you read this UAA, and you stop. So frame shift mutations are generally very deleterious because any downstream of the frame shift, it's just kind of reading the out of frame, and so you make a protein that doesn't make much sense. And you often hit a stop codon by accident because of that. So frame shift mutations are more deleterious usually than a missense mutation or a base substitution. Okay, so the other kind of thing I mentioned that causes mutations is transposable elements, and bacteria, all bacteria pretty much have transposable elements. And a transposon is a sequence of DNA that can produce a copy of itself, which can then insert somewhere else in the DNA. Okay, so these were discovered some years ago, first in corn, but they exist in bacteria as well. And E. coli has a number of these, and other bacteria have them as well. And so this is happening at some low frequency and can lead to spontaneous mutation. And it's kind of related to like a retrovirus. A retrovirus can integrate into the genome of animal cells. The difference here is it makes an RNA copy, leads to a DNA copy, which can then insert. So here there's no RNA intermediate, it just makes a DNA copy, which inserts. So it's kind of related. And when this copy inserts somewhere else, it's if it inserts in the middle of a gene, it would disrupt that gene. Okay, so mutations, you have spontaneous mutations, and then you can have induced mutations. And in the laboratory, you're always working with induced mutations, or generally you're trying to speed up the mutation process so you can get a mutant. And a mutant is really how you try to understand something. If you're studying humans, you have to wait until you get somebody that has a defect in their genome or a change in the genome that results in a phenotype, and then you study that. You try to find out what was the gene that's responsible for that phenotype. So in the laboratory, you can induce mutations, and then you get a mutant that has a defect, and then you try to find what the gene that's affected. So mutants are really important to understand, biology. And so you can increase the rate of mutations, and of course this can happen in the environment too. Chemical mutagens can cause the increase in mutation rate. Various chemicals cause mutations. Transposition, which is a transposon, and also physical, like radiation, like ultraviolet light, X-rays, these all cause mutations. Okay, so let's take a look at ultraviolet light, which is a frequent cause of mutations, because we're exposed to sunlight all the time. And what ultraviolet light does is it gets absorbed by the DNA, and where there's two pyrimidines next to each other, in this case it's two thymines, the ultraviolet light can cause a cross-link between these two thymines, and that kinks the DNA because it's abnormal conformation here. And then when the DNA polymerase comes on the other, it tries to copy that, it gets stuck here because that distorted structure cannot be readily copied. So thymine dimers or cytidine thymine dimers distort the DNA and cause problems. Other radiation, X-rays and gamma rays, can break the DNA, and they cause double-strand breaks, and these can be, in bacteria, they're often lethal. And you can have chemicals that, like base analogs or alkylating agents. Alkylating agents are agents that add a methyl group or an ethyl group to the DNA that can cause mispairing. So there can be all these chemical modifications, and then there's certain chemicals that can insert they're hydrophobic planar molecules that can insert between like two thymines and sort of push them apart and make the DNA look a little bigger than it is. And then lastly, you have enzymatic error. So the DNA polymerase is a pretty great enzyme. It copies DNA. It makes a mistake 1 in about 10,000 times, which is pretty low, but it does make mistakes. So let's look at some of these mutagens. So nitrous acid is a mutagen that leads to base substitution. So here, like cytosine, in the presence of nitrous acid, it gets converted to uracil. Uracil is not normally in the DNA, but when it's in the DNA, it looks like thymine, or base pairs like thymine. And so instead of C base pairing with a G, U would base pair with an A. And so that's going to lead to a base substitution. And then alkylating agents, there's lots of these kinds of chemicals around. They can add, like I say, a methyl group or an ethyl group to the base, and then they will alter the hydrogen bonding properties of the base. And so nitrosoguanidine, that's a very one that's used in the laboratory, causes high frequency of mutations because it methylates so many bases. Okay, so here's an example. So here's an example, like here, you have a normal base, and a bromouracil is a chemical that looks a lot like, it looks like thymine, but instead of base pairing with an A, it base pairs with a G. So 5-bromouracil, if it was added to a culture, it would get incorporated into the DNA, and it has a higher chance of base pairing with the wrong base, and so you introduce a mistake. And the same thing can happen with 2-aminopurine. It changes the base pairing properties, and so it will also lead to mispairing. So base analogs are just compounds that get incorporated into the DNA, and their base pairing isn't as rigid as the normal bases, and they base pair with the wrong base some of the time, and so then you get mistakes in the opposite strand. Okay, so the other kind of molecules that can cause base mutations are intercalating agents. And they, as I said, they're kind of hydrophobic planar molecules, and they insert between the bases in the DNA, and they create a space between these, and then an extra base, when it gets copied, sometimes an extra base can get put in there, and so you have an extra base, and it leads to a frame shift. So ethidium bromide was one example. Ethidium bromide is a dye. It's actually used to stain DNA because when it binds to DNA, it changes its fluorescent properties, and so it's used as an agent to stain DNA. But it's also, because it changes its fluorescent property when it inserts between the bases, but it also then can cause mutations when that DNA is trying to be replicated. Okay, so, and then I already mentioned transposons. These are used a lot in the laboratory because you can have a vector that carries a transposon. You can insert it in. You can deliver that vector into the bacteria, and then that transposon can jump from the vector onto the chromosome, and then it'll insert somewhere in a gene, and it can disrupt that gene, and so it usually causes an activating or knockout mutation of that gene. So in this case, you have the transposon here, 1, 2, 3. It jumps from this DNA molecule into this DNA molecule. If it jumped into gene B, then gene B would be disrupted because it would have this big transposon stuck in the middle of it. Okay, so to fight all these potential mistakes in the DNA, the cell has a bunch of DNA repair mechanisms. And this is to fight not only the spontaneous mutation that occurs from DNA replication errors, but also from all the chemicals in the environment that a cell could get exposed to that would cause DNA damage. And so you basically have several different kinds of repair. You can have direct repair where the damage just gets reversed. And that's very faithful. It doesn't lead to any mutation. You can also have an excision, and this is like for UV dimers. They get excised from the DNA, and then you just fill in the missing bases, and so that's also very faithful. And you can have recombinational repair if the chromosome is replicated and one gets damaged and the other one doesn't, you can have recombination between them, and then that can repair it. And then as a final sort of possibility, you can have this in bacteria, something called SOS system, which can repair the damage, but it tends to be not very faithful, and so it introduces errors. And it's called translesion biosynthesis because this polymerase works across the lesion and puts some base in there and can make mistakes. And we'll talk a little bit more about that. So that occurs when you have extensive damage. And it turns out that these translesion polymerases that exist in bacteria also exist in our cells. And so this is a way to respond to extensive DNA damage. Okay, so base substitutions, that they can occur, as I said, by DNA polymerase itself. And DNA polymerase has a function which is called proofreading. So when it copies a piece of DNA, if it puts in the wrong base, it can stop and back up and remove it and then proceed. And that increases the fidelity about 100-fold. So the polymerase makes a mistake 1 in 10,000, but it has this proofreading function, so now it's 1 in a million. And then, so that's pretty low, but you have 4 million, E. coli chromosome has 4 million base pairs, and so it could still make 4 mistakes every time it copied the genome. But then you have this additional mismatch repair. And mismatch repair is a system that can sense the mismatch in the DNA, and this perturbs the structure a little bit. And what it'll do is it can tell which is the old strand and which is the new strand. And in the case of E. coli, the old strand will be methylated, and once the DNA is copied, the new strand is not methylated right away, so it knows that's the new strand. It'll excise this mismatch and then replace it. And so that increases the fidelity another 100-fold. So now instead of making chance 1 in 10,000, 1 in a million, you're 1 in a billion almost. So you really can now very faithfully replicate the DNA, and very low chance of introducing mutations by polymerization. So now, an example is, I mentioned ultraviolet light causes thymine dimers. Now, thymine dimers kink the DNA, and so this causes a larger distortion of the DNA than just a mismatch. And so there's a special system in the cell and it's called UVRA, ultraviolet A, B, and C. And so it's called this because it was discovered for repairing thymine dimers, but it can also repair other large distortions in the DNA, and what it does is it's able to remove the part of the DNA that's responsible for the distortion. So it has these three proteins work together to remove this distortion. There is another mechanism, photoreactivation, which just reverses the thymine dimer. That can work if the bacteria is exposed to visible light, but if it's not, then it has to use this system. And this system is a little bit like the mismatch repair, except that it's dealing with bigger distortions, and so it also recognizes the distortion and then makes nicks on either side, removes the damage, and then repairs it. And so again, this is pretty faithful repair, and so it can remove the thymine dimer, and you won't have any negative consequence. Now, the problem that encounters is if you can introduce, like ultraviolet light will induce damage in the DNA, and so this system is trying to repair it, but if the replication fork is coming in and hits the thymine dimer before it gets repaired, then that causes a problem, because once the DNA polymerase hits the thymine dimer, it doesn't know what to do with that thymine dimer. It doesn't know what to put on the opposite strand. And so then the replication gets stalled. You know, if you have a little bit of DNA damage, they probably all get repaired before the DNA polymerase hit the damage. But if you have lots of damage, it's unlikely you would repair it all before the DNA polymerase would hit one of those. And so if the DNA polymerase hits one of those, then another system kicks in, and it's called SOS repair, SOS kind of for help. And it's an effort to bypass the DNA damage. So if you have lots of DNA damage, this system gets induced, a new polymerase gets produced, and this is a translesion bypass polymerase, and presumably it doesn't have as high a fidelity, so when it sees that thymine dimer, it can put something in there and then move on. And because it's not very faithful, you're going to get mutations. Okay, so this system is another regulatory system in bacteria, and it's responsible for most of the mutations, induced mutations in bacteria. And so what happens is this system produces a low amount of a protein REC-A, which we're going to come across again, it's because it's involved in recombination. And this protein binds to single-stranded DNA, and that's a trigger that there's something amiss in the cell and that there's DNA damage. And so what REC-A bound to single-stranded DNA does is it gets rid of a repressor called LEX-A, and this repressor controls a bunch of genes, including itself, REC-A, and then a bunch of DNA repair genes, including the UVR genes. So when you have DNA damage, you destroy this repressor, and all these genes get turned on because RNA polymerase can get to those promoters. And you produce more of UVR, A, B, and C, and more of this UMU, D, and C, which code for a new polymerase. Okay, so REC-A, which is involved in recombination, we'll see later, as I said, is really the sensor, because when it binds to single-stranded DNA, then it has the ability to inactivate the LEX-A repressor, which is the repressor that controls all these genes involved in DNA repair. Okay, so what happens when a DNA polymerase is copying this DNA with a thymine dimer in is it doesn't know what to put in here, and so you create a region of single-stranded. REC-A binds, and then when REC-A is bound here, this new polymerase is recruited, and the new polymerase then is able to, because it's not very faithful, it's able to put something in here where the thymine dimers are, and then it can move along and fill this single-stranded region, and it can make some more mistakes in here. So the idea is you can rescue DNA replication by making some mistakes by using a low-fidelity DNA polymerase, and then you're going to introduce mutations, though. But it's better than not replicating the DNA. Okay, so So these are the kinds of things that, all cells are exposed to, all these agents that can cause mutations. And mutations can give a rise to resistance to antibiotics, as we saw with like quinolones, or it happens also with some other antibiotics. And then these resistant genes then can be spread from one bacterium to another, or a cell that has that resistance gene can be selected for. But if it's going to be spread from one bacteria to another, then it has to undergo genetic exchange. And there, as we mentioned yesterday, there's three main pathways that bacteria have for genetic exchange: transformation, transduction, and conjugation. And it turns out that bacteria are very promiscuous in exchanging DNA. So not only is one E. coli strain exchanging information with other E. coli strains, E. coli could also exchange information with Salmonella. And even strains that are less related can transfer some kinds of DNA, especially these pathogenicity islands can be transferred between bacteria that are even more distantly related. And as we mentioned yesterday with MRSA, this can have a profound impact on virulence because one of the pathogenicity islands there encodes resistance to methicillin. But there's a lot of other islands that are there and can affect the virulence properties of methicillin-resistant Staph aureus. So we have this genetic exchange, and so if we look at the general features of gene transfer in bacteria, it's unidirectional. You go from, one of them is a donor, the other is a recipient. When the DNA, a piece of DNA gets transferred to the recipient, then you have two copies of that gene generally. And so it doesn't transfer the whole chromosome. Usually a piece of the chromosome gets transferred. And so it has two copies of that gene, and then it can undergo genetic exchange or recombination. And as I mentioned, for a lot of things, gene transfer is among more closely related, but it can also occur at a lower frequency among less closely related organisms. So let's look at transformation. So transformation is just the uptake of DNA from the environment. There's no, it's not present in a virus particle, or it's not present in another bacteria. It's DNA that's free in the environment. And this is a mechanism that occurs among many bacteria pathogens, although conjugation is probably the most prominent. But this is still a very efficient mechanism that some bacteria have for undergoing genetic exchange. And so the recipients have to be competent in order to take up DNA. So bacteria, when they're normally growing, they're not competent, but they can acquire the ability to take up DNA. And this usually happens when they reach stationary phase. So remember, they go to exponential phase, they stop growing in stationary phase. This is generally when competence gets turned on. So it's an example of this quorum sensing. It gets turned on when the bacterial density is real high. And transformation was demonstrated a long time ago by Griffiths, who was working with pneumococci, and what he observed is that if you have strep pneumo, it exists in two forms: the virulent form, which had a capsule, and the avirulent form, which was a non-capsule. So if you put if you heat kill the virulent form and put it inside the mice, there would be no effect. But if you didn't heat and activate it, of course it's live, it would kill the mouse. And if you had the non-capsulated form, you could put it into the mouse and it would have no effect. And then he did some mixing experiments, so he would mix the virulent form, the heat-killed form, with the non-virulent form, which was not heat-killed. And then he could rescue, this combination would kill the mouse, and he could rescue live virulent bacteria again. In fact, then he could just take the heat-killed wild-type bacteria, or the virulent form, and just extract the DNA, and he could just mix the DNA from here with this non-virulent form, and he could recover the virulent form again that would kill the bacteria, or kill the mouse. So what is going on is is that the avirulent bacteria is taking up the DNA. So when you have a population of bacterial cells, some cells are dying and lysing and releasing DNA, or if you heat and activate it, some of the bacteria are killed and the DNA gets released, and the bacteria become competent, they can take up this piece of DNA, and then this DNA can undergo recombination. It has to find the region on the chromosome where it's homologous, and then it can undergo a genetic exchange, and so this red piece of DNA replaces the green, and so now you have this sort of situation. So if, for example, here was a mutant gene for the capsule, this was the wild type, so this organism couldn't produce capsule because it had a mutated gene. Now the wild type gene coming from this organism could replace that, and now you regenerate the wild type organism that can make capsules. So this is just one example of how you undergo transformation. So the hallmarks of this is the recipient cell picks up naked DNA. This naked DNA has to undergo homologous recombination that involves REC-A. So REC-A is this recombination protein, which is also, as we mentioned, senses DNA damage. So it's a multitasker. And it replaces the homologous gene on the chromosome. So, and if they were both wild type, it would undergo this recombination, there'd be no effect, right? You wouldn't even see it. So you only see it because if one of these was different, and this difference conferred some phenotype that you could select for. So natural transformation, which is what we're talking about here, occurs among some bacteria, and these bacteria cells have to be competent, and the competence involves them having the machinery to take up DNA and then integrating these large fragments once they get inside the cell, recombining them onto the chromosome. And I mentioned competence is not, bacterial cells when they're growing exponentially are not usually competent, but they become competent when they hit stationary phase. So just to think about the DNA that's getting taken up by the bacteria. So here's a DNA fragment that gets taken up. If it's just a piece from a piece of chromosome from somewhere, it won't have an origin. It's linear. It just exists, and if it doesn't undergo recombination, it'll just persist there for a while, and eventually it gets degraded. So it just disappears. So the only way it has an impact, if it finds the homologous region on the chromosome and undergoes this recombination, it replaces the homologous fragment, and then it becomes part of the chromosome. So it has to get inside the cell, then it has to find the homologous region and undergo recombination. So the natural transformation has a number of stages. And so first the DNA has to bind to the surface. So here's an example. If it was like a piece of DNA that could confer streptomycin resistance, it has to get taken up by the bacteria. And the way transformation works in all bacteria is only one strand gets taken up. So here we have, in this case, the chromosome is indicated by these blue lines, and this is the gene here encoding streptomycin sensitivity. So now you have this piece of DNA coming in. It happens to encode streptomycin resistance. It comes in. It can undergo genetic exchange here. It replaces this part of the chromosome. And when it gets copied, this chromosome gets copied, this one will be streptomycin resistance. Okay? So the DNA binds to the surface, but only a single strand enters. It finds the homologous region. It integrates there. And then the mismatch repair could occur. One or the other would get changed, but if the resistant one survived, then it would encode resistance, and that bug then will be resistant to streptomycin, and if streptomycin is in the environment, then that bug would be selected for. And if there was no streptomycin in the environment, that bug would have no advantage, and it might occur at some really low frequency, but you wouldn't see it. It's only when you add streptomycin and you kill the 10 to the 10th bacteria, streptomycin sensitive, the resistant one then takes over the population. So there's a little difference between those organisms that can do transformation, whether they're gram-positive or gram-negative. And then, like I say, not all bacteria can undergo natural transformation, but a significant number of pathogens can, so we have to be concerned about it. So the way they become gram-positive is there's a buildup of a competence factor, which is an example of quorum sensing. So the more bacteria you have, the more of this competence factor you have. This stimulates the bacteria to become competent, and when they do, they have the ability to bind double-strand DNA. And in gram-positive bacteria, they bind any DNA, whether it's homologous or non-homologous, and they uptake this DNA, and then a single strand enters the cell, and then this DNA recombines with the chromosome by homologous recombination. So it has to be homologous in order for it to recombine. Now, in gram-negative bacteria, they're a little bit more choosy, and so they only take up DNA from closely related species because there's a sequence in the DNA that they recognize, and that sequence is unlikely to be present in bacteria from different genera or more distantly related. So they bind this DNA in a structure that's called the transformosome. It's on the surface, but it's much like gram-positive, except that it has specificity. So it recognizes the sequence motif, it takes the double-stranded DNA up, and again, a single-stranded DNA is then brought into the cell, and it has to undergo homologous recombination. So transformation is just this genetic exchange mechanism where bacteria just take DNA up from the environment, free DNA that's out there in the environment, and free DNA would come from cells lysing and releasing their DNA. And a lot of antibiotics cause bacteria to lyse, especially beta-lactams. That would release DNA. Other bacteria could take up that DNA and have the ability then to undergo transformation. So transformation, like I said, is maybe not the most prevalent kind of genetic exchange. We think conjugation is, but it's still important in some pathogens like Neisseria and Streptococcus and a few others. So it is an important mechanism of genetic exchange. So here's a question. Transformation differs substantially between gram-positive and gram-negative bacteria in which of the following characteristics? And so here's the options, and so you should be able to answer this just directly from the lecture notes, right? So. A. Right. Thank you. Okay, so we'll end there unless there's any questions. And tomorrow we'll finish up in talking about the other mechanisms of genetic exchange and a little bit more about plasmids and their contribution to antibiotic resistance.